A pleasant good morning to you all. Welcome to the Department of Informatics and Health Data Science webinar series entitled The Future of Health Data Innovation. My name is Azizi Seixas. I'm the interim chair for the Department of Informatics and Health Data Science, and I am extremely happy and enthusiastic about our second year of doing this incredible seminar series to share a little bit about who we are as a Department of Informatics and Health Data Science. We are a fantastic group of individuals. We have over 20 primary and secondary faculty who are really trying to transform healthcare as well as population health across 5 core verticals. This includes education, research, clinical care and operations and dissemination, as well as venture and service outreach policy and ethics. And so this webinar series, which for the first time we are now offering professional credit, allows us to build a community, not just here at the University of Miami, but locally and regionally and globally. And so I am so happy that so many of you have decided to join us today. There are folks here on the Zoom, and I know there are other folks as well who are joining us on YouTube as well as LinkedIn Live. And so I welcome you, and I hope that you will come to many more of these webinar series, as well as all the other educational activities that we have going on here. This could not be possible without an amazing cast of leaders in our department. And I would like to welcome one said leader, Dr. Mairead Maloney, who has done incredible work to put this fantastic program together. So without that, I know you've come to listen to the special speaker as well as Dr. Maloney. I would like to welcome Dr. Maloney to invite and introduce our speaker for today. Thank you so much for being here, folks.
Thank you, Azizi, for that incredibly kind introduction. I have the great pleasure of introducing Dr. Rebecca Shafee for us today, and I know we are all super excited to learn from her. But before I give her bio, I want to introduce you to our newest team member and a real VP, Alex Gonzalez. And Alex is going to share with you how you can claim CME credit today, and he'll also post it in the chat. I mentioned we have the great pleasure of having Dr. Rebecca Shafee with us today. She is a postdoctoral research fellow in the section on developmental neurogenomics at the NIMH, where her research focuses on understanding the biological basis of sex differences in brain structure and function using neuroimaging and statistical genetics techniques. Rebecca's academic training is in physics. She received her BS from Caltech and her PhD from Harvard University. Her PhD thesis tackled the problem of estimating how fast black holes spin. How cool is that? At the postdoctoral level, she transitioned to research in neuroscience as a Swartz Fellow, focusing on neuroimaging at Harvard, followed by further training in statistical genetics in the Broad Institute. Her current and past projects combine large, complex data sets with cutting-edge neuroimaging and statistical genetics techniques to investigate the genetic basis of brain structure and function in health, as well as disease. We're so proud and delighted to have her here today. And with that, I'll let her take it away.
Thank you so much for the kind introduction. I'm delighted to be here. It's an honor to be part of this very exciting seminar series. Let me share my screen. Okay, so just to tell you briefly, this is going to be the outline for the talk. First, I will talk a little bit about my research background and interest. Next, I will focus on a project that I recently completed at the NIMH, which I think shows how large data sets can be used to probe interesting biological questions with medical relevance. And lastly, I will tell you a little bit about some projects that I have in mind that I would like to work on in the future. Dr. Maloney has already told you some of this, but I just want to emphasize that I've had a somewhat unconventional path to neurogenomics. I was born and brought up in Bangladesh, and then I moved to Southern California to study physics at Caltech. And then I continued physics at the graduate level, had my PhD at Harvard, where I measured the spin of black holes. But near the end of my PhD, it became obvious to me that I wanted to focus my research on biological questions. In particular, the brain was very intriguing. And so I took advantage of a fellowship that was being offered at that time that allowed people who had quantitative backgrounds to kind of jump into neuroscience without much knowledge of neuroscience. With Dr. Randy Buckner's group at the Center for Brain Science and did a lot of neuroimaging work. And I thought it was fantastic that we could use MRI to look inside the living brain. But at the same time, it was also becoming obvious to me that we also need to understand the genetic basis of how the brain works. So I continued my training at the Broad Institute, where I led multiple projects using statistical genetics methods on large biobank scale datasets. And currently, I'm at the NIMH in Armin Rozan's group, where I get to combine my passion for the brain, as well as statistical genetics, and also for big data, where I get to apply all of this to the projects that I'm working on right now. Now, here is an example of my papers. So let me tell you a little bit, not a lot, a little bit about each of those. So this is my first paper, and this is a paper where we reported the spin of 3 black holes in our galaxy. And we did that by developing, by looking at these binary systems in our galaxy, which consists of a star here and a black hole, which is at the center of this disk. And as the gas from the star is being kind of eaten up by the black hole, it gets really hot and forms a disk around it. And the hot gas emits X-ray. The X-ray telescopes detect the X-ray, and the grad students sit in front of their computers and they can model that X-ray to estimate how fast is this black hole spinning. And we were actually the first group to report the spin of the black holes in our galaxy. So it was quite exciting. But then I kind of switched my research focus. So this is a paper I wrote as a neuroscience postdoc at the Center for Brain Science, where we developed a method of looking at the cortical myelination pattern using MRI imaging. So myelin is a very important part of the brain, and it changes over one's lifetime. And as well as abnormal myelination has been linked to disorders such as schizophrenia and multiple sclerosis. So what we did is we found a method, we developed a method to measure the myelination pattern of the cortex, also incorporating corrections for the fact that these MRI images are millimeter resolution scale. So next, we applied it to about 1,500 brains of the age range 18 to 35 years. And on the left here, you see the group average map of cortical myelination. So the regions that are in yellow and red, the motor cortex, visual cortex, these are the regions that are highly myelinated, whereas the blue regions, such as the prefrontal cortex, don't have as much myelination. And this was consistent with postmortem study that actually looked at the brain tissue directly to assess myelination. In addition to other things, we showed that in this cross-sectional data set, even within 18 to 35-year age range, cortical myelination kept increasing. And at the time of publication, our paper was, basically this was the largest reported study of in vivo cortical myelination of the human brain. Now, at the bottom, these 2 papers are more recent. They came out last year, and they're more representative of what I'm interested in right now. The one here, shown in red, this is a paper on a project I worked at the Broad Institute. Some of you are probably familiar with the UK Biobank, which is a large population biobank and has thousands of phenotypes that are health and disease related. And of course, it's a very, very useful data set, but at the same time, when there are thousands of different items, they're also correlated and overlapping, and it's hard to work with these individual thousands of measurements. So what we did in a large, highly collaborative project that spanned several years is we reduced these thousands of phenotypes to a handful of 35 factors that are easier to interpret. I will talk more about this project near the end of my talk. And here on the bottom left is what I'm going to talk about mostly today. We looked at the sex difference in the genetic basis of the brain anatomy in the UK Biobank data. So for this part of the talk, first, I will try to motivate you that this is indeed an interesting question. And next, I will give you very brief statistical genetics primers so that we can understand the findings that I will talk about later. So to look at the living brain, we use MRI, or magnetic resonance imaging. What MRI does is it takes pictures of the brain vertically. So imagine cutting the brain vertically through the center like this. And the image on the left is basically the MRI equivalent of what you would see. And with state-of-the-art softwares, now it's possible to reconstruct an individual's brain on the computer and also measure different things. For example, how thick is this person's cortex at this specific location? How large is this person's cerebellum? Such things, and then they can be used for research. So the first motivation is brain structures are heritable. What does heritable mean? It means that a significant fraction of the variation that we see from person to person can be explained by genetic variation. So what I'm showing you here is a figure from Thompson et al., 2001. So it's one of the earliest papers with brain MRI images. And what I'm showing you here is the correlation in gray matter volume on the surface, shown on the surface of the cortex between pairs of twins. So on the left, we have identical twins who basically share all their genetic material. And on the right, we have fraternal twins who are like siblings, and so they roughly share half of the genetic material. And what we see here is that the colors here are that pink and red indicate high correlation and blue indicate low correlation. So we see that across much of the cortex, the correlation between the identical twin pairs is much higher compared to the correlation in the fraternal twins. So this is an example of a trait where genetics play an important role. It's an example of a heritable trait. Now, the second motivation is that many studies over the years have established that there are significant sex differences in the anatomy of the brain. For example, males have larger head size and larger total brain volume, which may not be a surprise to us because on average, globally, males are taller than females, about 10 centimeters. So somebody who's taller also has a bigger head, also has a bigger brain. But even if we control for height and head size, even going beyond that, we can still see significant regional sex differences. So this is a figure taken from a paper from our group, which shows the sex difference in gray matter volume using 488 males and 488 females. And the regions that are highlighted in blue, these are female biased, which means these regions have higher gray matter volume on average in females, whereas the regions that are shown in orange are the regions that have higher gray matter volume in males. In fact, these sex difference of gray matter maps are quite reproducible across different data sets. So this is a figure taken from Decation et al. On the left is the figure I just showed you. So this is the HCP data set. In the middle is the same analysis done in the UK Biobank data. And on the right, I'm showing you results for the same analysis done on the Lotz et al. Paper. So these are three distinct data sets. And if you look at the bottom panel that shows the brain surface, you see that even just by looking at it, we can tell that there's a high level of similarity and reproducibility for this sex difference pattern of gray matter volume. Now, if these phenotypic sex differences could potentially reflect influences of sex-specific biology on brain development. For example, take this figure from Wright et al. That shows that testosterone influences the volume of the preoptic area of rodents, but only in males and not in females. So you can imagine that if there are genetic variants that influence the strength of these sex-specific biological processes, then those variants might also be detected as producing sex-biased effects on that specific brain feature. The third motivation, which I think many of you are aware of is that there is significant sex difference in the prevalence of neuropsychiatric as well as neurodevelopmental disorders. This figure shows the relative prevalence of multiple disorders in the population. And we see that while autism and schizophrenia are male biased, we see that depression and bipolar disorder, for example, these are female biased. And not just the prevalence of these disorders, there's also sex difference in how the disorders manifest themselves in the brain. This is a paper by Guma et al. That looked at the thickness of the cortex in control groups and compared it to individuals who are at high risk for psychosis. And what they found is that in this region, that is marked with an arrow here, this is the postcentral gyrus, that the females that were at high risk actually had thinner cortex compared to the controls, whereas the males at high risk had thicker cortex. So this is an example of a sex difference or sex biased effect, manifestation of this specific psychiatric risk. Now let's move on to genetic variation. When I'm talking about genetic variation, I mean, I'm talking about a specific type of variation. These are single nucleotide polymorphisms, or SNPs. So if you look at this figure, in the top row, this is person 1. And if you focus on this gray region, you'll see that this person has a C nucleotide at this region, whereas person 2 has the T nucleotide. So we have chromosomes in pairs. So in the population, individuals can have one C and one T, or two Ts, or both C alleles. So these are the possible genotypes for this specific SNP. There are roughly a few million SNPs in the human genome. And with current technology, it's possible to use an individual's blood, for example, and with the help of these genotyping chips, we can kind of measure all the SNP variability for individuals. So the data that we work with in statistical genetics looks something like this. So basically the different alleles for each of these SNPs for each person. So it can be like millions of SNPs versus 100,000 people. Pretty massive data sets. And let me now talk about phenotypic variation. All this genetic variation, for example, coming from SNPs, they cause phenotypic variation. And most physical traits, such as height or weight or BMI, these are all polygenic, which means that many genes contribute towards the variations of these phenotypes. So if we take height, for example, let's say this is the distribution of height in University of Miami. It's approximately normal, and it has some variance, right? And so on the left, here's a cartoon that shows all our chromosomes in pairs. And let's just say that all the V's here mark basically all the SNPs in the human genome. But the ones that are shown in orange are the ones that somehow influence height and introduce the variance we see in the population. So the goal of statistical genetics techniques is to identify these orange SNPs that are relevant for a phenotype, and to also estimate their effect, individual as well as all of them together, their effect on the phenotypes. Now, here's the statistical genetics primer. There are three concepts that are relevant for the paper I'm talking about. The first one is GWAS. GWAS stands for Genome-Wide Association Study. So once again, let's go back to a single SNP. This SNP has two alleles, T and C, and in the population, we have these three genotypes. So let's say I want to find out if this SNP does indeed influence the phenotype of choice, which could be height. So I plot the count of the T allele, which can be 0, 1, or 2, versus the phenotype. So each dot here is an individual person. And what we see is that in this case, the T allele indeed increases the value of this phenotype. So this is an example of a significant GWAS hit. And so when one does a genome-wide association study, this is repeated across all the SNPs. And the result is something called a Manhattan plot, which is shown on the right. And this Manhattan plot shows the X chromosomes, and then each dot basically is an individual SNP, and the y-axis is the strength of association with that phenotype. So we see there's a big peak here. So a bunch of SNPs on chromosome 3 seem to be associated with this specific phenotype. But what happens is that in the case of these SNPs, for complex traits like height, you'll find thousands of SNPs that are associated with these phenotypes, but the effect sizes are small. So with the availability of large biobanks and international collaborations, it is not possible to run statistically powered GWASs on many brain phenotypes. So this is a work from Grasby et al. From 2020. And what they did is they combined 35,000 brain MRI images across two different data sets, and they ran GWAS on brain phenotypes. They divided up the brain into these regions that are shown in different colors, and they measured the surface area and cortical thickness for each of these regions, and then GWAS for each of these phenotypes. On the left is a beautiful diagram that shows you all the GWAS hits across all the chromosomes, where the colors correspond to the color of the brain region, and basically the shape corresponds to what type of phenotype that was, whether it was surface area or cortical thickness. And what I find beautiful about this figure is how nicely it shows us the polygenic nature of all these brain traits. The second concept is SNP heritability. So SNP heritability means we want to understand how much of the variation in a trait, so this variance that we see when we plot a trait, how much of this is coming from common SNPs only. So it's the contribution from the SNPs. So if we divide the phenotypic variance as a sum of genetic variance and environmental variance, then H squared, the heritability from SNPs, is defined as a ratio of the genetic variance to the phenotypic variance. And this can go from 0 to 1. In the case of 0, we're basically looking at a phenotype where the genetic variance has no contribution to the phenotypic variation. Whereas if we have a heritability of 1, that means the entire variation that we see in the phenotype can be explained by just the SNP variations, which is never the case. So usually what we see is that most of the brain phenotypes are somewhere in between 0 and 1. But how does one estimate SNP heritability in a population data, right? So a common approach is to use a software called GCTA, in which we use the genotype data to construct genomic relationship matrices. So even when we are working with unrelated individuals, some people are genetically more similar compared to others, and that's the information that the genomic relationship matrix contains. And this matrix can be used together with some phenotype, that what the heritability of that specific phenotype is. And this is one way of doing it. And an alternate approach, when people don't have individual level genotype data, is to use the summary statistics from GWASs and to just use that to estimate the heritability. This is one of the earlier papers that looked at the brain phenotypes from UK Biobank in about 8,500 individuals. And what I'm showing you here is the SNP heritability for a lot of thousands of different phenotypes. There are 3 different panels, the top one corresponding to structural MRI, the middle one to diffusion MRI, and the bottom one to functional MRI. And even within each category, there are different colors corresponding to different types of phenotypes. So the purple ones here are volumetric measures. The light green ones here on the top panel, these are cortical thickness measurements. And we can see that there's a wide range of distribution of phenotypic heritability. And on average, it looks like the functional MRI measurements are the least heritable compared to the structural MRI and the diffusion MRI phenotypes. Now, the third concept is genetic correlation, denoted by RG. We're all familiar with Pearson's correlation. What does Pearson's correlation tell us? The similarity of 2 different variables. Similarly, what genetic correlation tells us is how similar are the genetic architectures of 2 different traits. So this can also go from -1 to +1. So if the genetic correlation is +1, that means the SNPs have very similar genetic effect on each of these 2 traits. Whereas if you have a negative correlation, that means that the SNPs actually have opposite sign effects for the 2 traits. Here's a really nice plot that shows the genetic correlation for many health-related traits in the UK Biobank. And so the blue color indicates positive correlation and red indicates negative correlation. You'll see there are some clusters here. So if we look at this big cluster here, this is waist circumference, hip circumference, trunk fat, weight, BMI. We know that these are all phenotypically correlated traits. So genetically, when we run GWASs, it seems like they're also genetically quite similar. At the bottom, there's a little cluster that consists of type 2 diabetes and hemoglobin A1C. We know that those two are related, so we can see that in the genetic correlation plot also. Once again, for genetic correlation too, one can use GCTA and individual level genotype data to calculate these correlations, or in the absence of individual level data, you can just use GWAS summary statistics using fantastic softwares like LDSC. Now, I earlier showed you the GWAS plot from Grasby et al. So the authors also looked at the genetic correlation between brain phenotypes and brain-related phenotypes. So what I'm showing you is the genetic correlation for total surface area and average cortical thickness with all these neuropsychiatric or brain-related phenotypes. So for total surface area, we see there's significant positive genetic correlation with educational attainment, general cognitive function, Parkinson's disease. What that means is that there are many SNPs that increase total surface area of the brain and also increase educational attainment, general cognitive function, and the risk for Parkinson's. On the other end, we see significant negative genetic correlation between total surface area and major depressive disorder, neuroticism, insomnia, ADHD. What that means is that there are lots of SNPs that are increasing the risk for these disorders, but at the same time, they're decreasing the total surface area. So that's what gives rise to the negative genetic correlation. So this was for the surface area of the cortex, but for average cortical thickness, the thickness of the gray matter of the cortex, the authors did not detect any significant genetic correlation. So even within the brain, even within the cortex, we can see how different anatomical features have different relevance for neuropsychiatric disorders. But all the studies that I talked about, they did combined analysis. They did not look at the males and the females separately. So now I will show you results from our project where we systematically screened for potential sex biases in the influence of common genetic variants on brain anatomy. And we do that using the UK Biobank data. So UK Biobank has over 500,000 people's genetic data, health-related data, but the MRI sample size is somewhat smaller. The current release has about 40,000 individuals. And after standard genetic and MRI QC, we were left with 30,000. And it's a slightly female bias sample. So we have 52% females and 48% males. And it's a somewhat older cohort. So the average age was 64 years. And within the MRI images, the phenotypes we chose were 3 global brain measurements. So the mean gray matter thickness, the total surface area, as well as the total volume of gray matter and white matter. And to look at the regional phenotypes, we divided up the brain, each hemisphere, into 180 regions. So a total of 360 parcels. And for each of these parcels, we basically measured the thickness and the volume, as well as the surface area for that region. And lastly, we also looked at 23 subcortical volumes. So this would be volumes of the amygdala, of the hippocampus, etc. And these were all corrected for age and other things that we don't care about, for example, a scanner site or head motion within each sex before doing the analysis. And here is the analysis pipeline. So we have millions of SNPs, and we use that to construct genomic relationship matrices for males and the females separately. So we have a male GRM and a female GRM. Then we combine it with the phenotypes that we have to estimate the heritability of each of these 1,100 phenotypes in males and females separately. And then we ask the first question, which is there sex difference in the heritability of these phenotypes? Next, we use the GRM to calculate genetic correlation for each phenotype between males and females. And what we're going after is there a phenotype where the genetic correlation is less than 1? Because that would tell us that there is difference in the genetic architecture between the sexes. And lastly, we ran sex-specific GWASs for all of these phenotypes, which means about 2,200 or so phenotypes and GWASs. And we looked at sex difference at the single variant level, at the single SNP level, and asked if there is any SNP that shows sex-biased effect on any of these phenotypes. So here are our results for SNP heritability. I'm showing you only the left hemisphere because the results were quite symmetric for left and right. And the left panel here shows gray matter volume, the middle one surface area, and the right one cortical thickness. On the top, I have females, and at the bottom, I have males. And these lines, the gray lines on the cortical surface, show you the different regions for which we assessed heritability. So the first thing we notice is that, also the colors. Dark purple indicates high heritability and white means low heritability. So the first thing we notice is that there's a wide distribution of heritability across the cortex for all of these measurements. And this is the visual cortex. The visual cortex area seems to have the highest heritability for both gray matter volume and surface area. The second thing we notice is that on average, gray matter volume and surface area seem to be more heritable compared to cortical thickness, which is shown on the right. And the third thing we notice is that the maps look very similar between the males and the females. And that becomes obvious when we make scatter plots. So in these plots, each dot corresponds to the heritability measure of male on the x-axis and female on the y-axis. And the color corresponds to which lobe of the brain that region belongs to. And what we found is that there's high correlation for gray matter volume, surface area, and cortical thickness when we look at this. So high level of correlation between males and females. And while at the individual regional level, we did not find any significant sex difference in heritability. When we pulled all the regions together within each category and did a paired t-test for males and females, we actually found that females had higher heritability for gray matter volume and surface area compared to males. That was not the case for cortical thickness, though. Now, the second set of results, this is the genetic correlation map on the cortical surface. Once again, is for gray matter volume, surface area, and cortical thickness. And here, high correlation is shown in is shown in white, and the low correlation regions are shown in dark blue or purple. And what we see that while there is some distribution across the cortex in the genetic correlation value, there are many regions where the correlation is close to 1. So in the formal test, where we tested for R less than 1, after doing that, this is what we find in the bottom. And this is showing the p-value of the test. We found that after correcting for multiple testing, only 2 regions out of these 1,000 phenotypes, only 2 regions showed significant deviation from a perfect genetic correlation. One was Brodmann area 5, and the other was posterior insula. But since one of the motivations for looking at genetic correlation was that anatomically we do see sex difference, and so we wanted to compare our genetic correlation map to the anatomical sex difference maps. So this is how sex difference looks like in gray matter volume, surface area, and cortical thickness. The regions that are red are higher in men, and the regions that are blue are higher in women. And what we found is that the 2 regions where genetic correlation was significantly less than 1 in the previous slide, those also happen to be 2 regions that are phenotypically sex biased. So Brodmann area 5 turns out that that is female biased, whereas the posterior insular region here, that happens to be male biased. But apart from those 2 specific cases, when we looked at this relationship across all the regions by making these scatter plots of RG against sex difference in the phenotypes, we found that for gray matter volume and cortical thickness, there was basically no relationship. Whereas for surface area, we saw some weak positive correlation. So the regions that had lower genetic correlation also tended to be more female biased. And as I mentioned, we also looked at subcortical volumes and global structures, global measurements. And on the left, I have the plot that shows you the female and male heritability for the global and the subcortical phenotypes. We did not find any sex difference there. And on the right, I'm showing you the male-female genetic correlation map for these phenotypes. And here too, you can see that all the correlations are 1. Basically, they're not detectably less than 1. So there wasn't much evidence of sex difference there either. And lastly, I would like to tell you about our results for single SNP level sex differences. So imagine I have the female GWAS and the male GWAS for a trait, one of the brain traits, right? And you can see that there are some regions here, for example, this peak here, where both sexes have that effect that GWAS hit, but there are also some regions where only, for example, the ones that I'm showing you with arrows, these are only seen in the female GWAS, but not in the male GWAS. So what we did is for each phenotype and for each SNP, we calculated a z-score of sex difference, which was basically the difference in the effect size of that SNP for that phenotype, and then converted that z-score to a p-value of sex difference. And this figure kind of captures all these results together. This was the one that took the hardest to make, the longest to make, because it was a lot of data and then putting it all together in this one figure. So on the left, I'm showing you results for the gray matter volume phenotypes. So 360 regions and all the SNPs. So each point is a SNP. And the x-axis shows you the different chromosomes. And for each SNP, I'm showing you the most significant sex bias p-value it had across all 360 phenotypes in that category. And I'm also showing you 2 different thresholds. The red line corresponds to the strict threshold that corrects for the total number of tests that we're doing. And the black line here, it only shows us the genome-wide significant threshold, which is the less stringent threshold and only accounts for how many SNPs we're testing. So what we saw is that for surface area and cortical thickness, no SNP survived the strict threshold. For gray matter volume, we found 2 SNPs that did make the strict threshold, and one of them was on chromosome X. And this SNP, however, was not within 100 kilobase pair of any gene, any protein coding gene, and it was not in any known enhancer or promoter region. But the one on chromosome 16, interestingly, was it was in one of the intronic regions of a gene called RBFOX1. So as it turns out, RBFOX1 is actually a gene that is expressed in both excitatory and inhibitory synapses. And it plays an important role in the brain development. When we looked at the expression level of RBFOX1 across different tissues using GTEx data, so each box here corresponds to different types of tissues, and red means high level of expression. We found that RBFOX1 is highly expressed in the brain, but there's also a little bit of expression in the skeletal muscle and in the heart. And literature search showed us that RBFOX is also associated with multiple neuropsychiatric and neurodevelopmental disorders, including ASD, schizophrenia, mood disorders, and ADHD. So because this SNP is interesting, we also wanted to take a closer look at its pattern of sex bias on the cortex. So I'm showing you the sex difference pattern for that specific SNP on this cortical surface in gray matter volume with a less stringent p-value threshold. And the orange regions are male biased and the purple regions are female biased. And this arrow here shows you the region from which that very strong signal came in the previous page, in the previous slide. And when we focus on that region, which is Brodmann area 6, what we see is that individuals that have the CT genotype for this SNP in males had higher gray matter volume compared to the TT genotypes. However, that was a male-specific effect because that was not seen in the females, and that is the source of the sex difference. When we lowered the threshold and made it so that more SNPs were available to work with, we were left with about 500 or so genes. And these 500 genes, so we were like, okay, let's try to figure out if they have any functional relevance. So we ran Go enrichment analysis, but none of the Go categories, functional categories, were enriched. So overall, we see high level of similarity between the sexes in the common genetic architecture of the human brain phenotypes that we have looked at. No individual brain region showed significant sex difference in SNP heritability. And this is something that has been found in other phenotypes too, actually. For example, it has been reported that systolic and diastolic blood pressure, as well as waist circumference, these are phenotypes that show higher heritability in females. We also found that between sex genetic correlation was not significantly different from one, with only 2 exceptions. And we also found that there wasn't much overlap with phenotypic sex difference and the sex difference in heritability and genetic correlation. And lastly, we saw that only 2 SNPs out of 12 million that showed statistically significant sex-biased effect on brain anatomy. This is consistent with something that was suggested by Zhu et al. In 2023. Basically, what they suggested is that the gene by sex interactions may largely act through sex differences in the magnitude of many genetic effects, so kind of an amplification effect, rather than having totally different causal variants or having opposite directions of effects. But as we know, the variations we see in the brain are due to genes as well as the environment and also the interaction between the two. What we have looked at so far is brain structure, but one can also look at brain function and behavioral and cognitive phenotypes. On the genetic side, we looked at common genetic variants, but we can also look at rare genetic variation. We need to understand how SNPs influence gene expression in tissue and cell-specific ways, or how epigenetic effects influence gene expression and hence brain structure. And on the purely environmental side, we have pollution, diet, stress level, activity level. These are all known factors that influence mental health condition. And this can also happen in a sex-biased way. So fortunately for us, complex data sets that capture the different dimensions of health are becoming increasingly accessible to researchers. UK Biobank alone has so much health and disease-related information available. I've mentioned neuroimaging and genotyping so far, but add to that socio-demographic data, lifestyle factors, other organ MRIs, for example, heart MRI, wearables, blood biomarkers. So basically, there are so many questions one could ask in addition to sex difference in the about the brain or sex difference or mental health. There are so many questions to ask. And I think these deeply phenotyped data cohorts are ideal for understanding neuropsychiatric and neurodevelopmental disorders, because these often show combinations of symptoms. If you look at this figure for autism, you'll see that there are some core symptoms that are shown in dark blue here, which are basically social deficits, language impairment, repetitive behaviors. But in addition to that, there are systemic associated issues, for example, GI disorder, immune dysfunction, etc. So we can explore the full spectrum of phenotypic outcomes associated with disease risk in diverse phenotypes in the biobank scale data sets. Let me walk you through a project that I have in mind. So I would like to understand the shared and sex-specific brain correlates, environmental factors, and genetic underpinnings of anxiety and depression. And for this, I would like to start with the results of our factor analysis paper. So let me just tell you very briefly what factor analysis is. It's a technique that is used to identify the latent relational structure among a set of variables, with the key assumption here being that if you have correlated phenotypes, it's due to some kind of a latent factor. So if you take this correlation matrix, for example, you see questions 1, 2, and 3 kind of cluster together, 4, 5, and 6 cluster together, and 7, 8, and 9 cluster together. So these would each of these clusters would belong to a separate factor. So here are the results from our factor analysis paper, where we reduced over 700 phenotypes to 35 factors. And the factors, the numbers on the left, the number actually tells you how much variance that factor explains. So while we see things like gastrointestinal issues, diabetes, eye conditions, the top 3 factors in UK Biobank are anxiety and depression related. So if you look at the individual components, this is what they look like. And you can see clearly there's a lot of correlation between these phenotypes. One would be expected given the other. So instead of using these phenotypes separately, it's more concise to just use the scores of each of these factors. So my plan is to use the factor scores for these 3 factors for each individual, and then also to calculate scores of the other factors, for example, chronic pain, or age-related eyesight deterioration. And then the correlation would give me shared or sex-specific environmental and health correlates of anxiety and depression. I could also use these anxiety and depression scores to find the neural correlates in a shared and sex-specific way by correlating these scores to neuroimaging phenotypes, for example, brain volume. And lastly, I could run GWASs on these anxiety and depression measures, also in a combined and sex-specific way to get the genetic correlates of anxiety and depression. And finally, we could try to find the overlaps in these findings to better understand the contributions from our brain, the genes, and the environment. So what I showed you here is for UK Biobank, which has an age range of 40 to 80, but I'm also interested in probing that the time of life when people are particularly in high risk for developing neuropsychiatric disorders. One of my long-term plans is to better understand the neuroanatomy and genetic basis of perinatal depression and anxiety. We know now that profound changes happen in the brain during pregnancy. Emily Jacobs' group has produced some fantastic papers that show that. And about 15% of women who give birth experience some form of perinatal, postnatal anxiety or depression. And at the present, the best predictors for that are still something like, okay, what is your mental health history or what is your socioeconomic status? But we know that these complications are to some extent heritable, and we know that there is overlap with major depression. So I would love to be part of a project that that follows expecting mothers through pregnancy and in the postpartum period, collecting neuroimaging, genotype, and other clinical data in diverse populations, and so that we can find better predictors of perinatal anxiety and depression. I know that already has the FemTech platform, which has been phenomenal in creating a platform for women's health. Perhaps I could be of use there. And here are the two people who motivated me to think about this specific project. So so I know all of you are interested in data. So there's a lot of data out there, health data, and they're complementary. Complementary means we can ask complementary questions with these data. For example, the age range is important when one is trying to look at neurodevelopmental problems as opposed to aging problems, or but a longitudinal versus cross-sectional. That can be an issue depending on what question you're asking. Also, biobanks have a lot of phenotypes, but often don't have a lot of patients' cases, which can be done with disease-focused data. So just an example here, UK Biobank, which is massive, only has 40 to 80-year-olds, and it's mostly European ancestry, whereas all of us, which was collected here in the US, is more diverse and has a wider age range. However, it does not have brain imaging. And ABCD is designed to be a longitudinal study. And so it's designed to understand how adolescent brains develop. So that has its own, its own problems, and one can ask so many questions there too. But these are all complementary data sets. And on top of that, collaborations are now on the way to integrate diverse human population data from around the world in trying to generate multi-ancestry genome-wide association studies of disease, which I think is essential for our understanding of the biology and the genetic basis of human complex traits and diseases. I think exciting science can be done by collaboration within a research field as well as across disciplines. And I'm excited to see the new findings that emerge in the coming years. With that, I would like to thank our team at the NIMH. So I work in Dr. Armin Raznahan's group. This is our fantastic team. Some of the members are wet lab people, some are computational people like me, some are clinical people. It's a beautiful combination and a wonderful, wonderful place to do research. Previously, I was at the Harvard Medical School and Broad Institute, and I had the opportunity to work with Randy Buckner, Steve McCarroll, and Ellis Robinson's group. Thank you.
Absolutely incredible presentation. Thank you so much, Dr. Shafee. We just have a couple of minutes, and I see that there are a tremendous amount of questions in the Q&A. I also want to take a second to remind folks to look in the chat to sign up for your CME credit. And I want to note that we have our next webinar on the 23rd of October. I'll post that momentarily. Dr. Shafee, to summarize some of the questions that I've seen coming through, just for the sake of time, if you could estimate for us, what percentage of brain structure do you think is heritable? And do you think certain parts of the brain, the structures are more heritable than others? And then as a secondary question to that, what factor do you think what percentage of the brain do you think is shaped by social factors and experiences?
I think that's a fantastic question. So let me answer the first part first because I know the answer. So basically, pretty much all the brain structures that you can measure at the millimeter scale, uh, distributed across the entire brain, are heritable to some extent. Now, the range of heritability can go from 0 to 1, but many of these structures are heritable or have a heritability of around 0.2, for example. That means only 20% of the phenotypic variation is due to the genetic variation that we can capture just with these SNPs. So the rest of it we don't understand, and we just call it environmental factors influencing. And so within the environmental factor, then of course there are social factors, but then there's also, what is this person eating? What is this person watching on Netflix? I don't know whether a kid is having too much screen time. So, there are so many different things. So the fraction of the phenotypic variance we see that's coming from environmental and social factors also varies across the brain. And it's high for some, low for some. So I think it's a great question. And I don't think we still understand the individual contribution for specific social and environmental factors on the brain very well, because even to detect a heritability of 0.2, we need a large data sample. So a lot of this is kind of a numbers game. We will detect smaller and smaller samples effects as we have more and more samples. Does that answer your question?
Yes, you did an amazing job. We'll go just maybe a minute or two longer with questions because again, there were so many and they were so excellent. Let me do, and I might go a little backwards. Susie Bianco asked, why would autism predominate in males while ADHD would predominate in females?
Yeah, this is a great question, and it is a very, I would say it's a very hot topic of research currently, both from just disease perspective as well as from understanding the genetic basis. Um, well, one kind of epidemiological explanation is that often it's more likely to be detected in males because females often hide it, so they're less likely to go to the doctor for the same symptoms in males. So that's one explanation, but I think there may be also sex-specific genetic effects that influence and may contribute towards the difference that we see.
Excellent, excellent. Maybe one more question. Brittany asks, are there sex differences in the extent of neuroplasticity capacity? And if so, what might be the reason for this?
That's also a great question. So I do not work at the cellular level, but this is my understanding that, even if we look at the functional performance or functional networks of the brain, there are some functions, there's some tests where men perform slightly better than women, and then there are some functions where women tend to perform better than men. And there may be some evolutionary route to that. We don't really know. And so given that, so that is something we're observing from the outside. The circuitry involved in that, you can imagine that there might be some sex bias in the neuroplasticity level that causes this overall functional sex difference that we see. So that's how I see it.
Really, really fascinating work. I wish we had time for all the questions. We will be, to answer someone's question, providing a recording and the slides so that people can go back and reference this. And of course, Dr. Shafee is I'm assuming available on email to answer
Yes,
questions as
I
well.
am.
Yeah, I'm going to let our fearless leader, Dr. Azizi Seychas, take us to the end here.
Oh, sure. Well, thank you so much, Dr. Shafee. That was an amazing, amazing presentation. And I just want to thank all of you as our audience for just being super focused and just intrigued by it. Because I know I was when I spoke to Dr. Shafee a few months ago. So thanks again, Dr. Shafee. Just an amazing, amazing, amazing presentation. And
Oh,
then
thank you.
also, of course, just super amazing. And we look forward to collaborating with you. We're doing some really cool things that I don't want to say too much right now. But you will hear more from and about Dr. Shafee in the near future because we're doing some really cool things together in our department. But I would definitely love to say thank you so much, Dr. Maloney. You have outdone yourself again, as well as Alex and LaRonda and the team. This has been an amazing presentation. I learned so much about the work that Dr. Shafee is doing, and I think it's incredible because we have so many wonderful programs going on here at the university in terms of human genetics through the Hussman Institute. We're going to be, the dean himself has invested over $30 million in neuroscience and aging and computational biology, of which, Dr. Shafee covers all of those areas as well. So this is, exciting times for us here at the university. And I just want to tell you that we have another amazing presentation next month, which Dr. Maloney dropped in the chat. And it's going to be a little bit different, but it's still focused on the future of health data innovation. It's going to be looking at the role of AI in radiology, but more so not just radiology, but diagnostics and imaging. And how is it that we can make healthcare more accessible, less lag time with the use of AI, while at the same time making it ethical. So stay tuned, make sure you register for that, and we will provide you more updates. So thank you all so much and have a wonderful day.
Thank you.